Hello and happy Tuesday, everybody. Today I want to talk about what I've been doing at the research lab I've been working at. This video will be divided into 3 parts. The first one is growing magnetotactic bacteria and how to do that. The second part is going to be about how I've been growing my plants to look at how magnetic fields affect root growth. The third part is about early steps in developing a technique for transferring that bacterial operon that helps make those little magnetic vesicles in the magnetotactic bacteria into plants and how I intend to go about that. The first thing I wanted to talk about was how to grow magnetotactic bacteria, and I think a good way to start talking about how to culture any organism is talking about its wild habitat. The habitat immediately behind me is a good example of the kind of habitat Magnetospirillum inhabits. In any wetland environment like this, you'll have a gradient of oxygen concentration with high oxygen concentration near the surface of the water and lower oxygen concentration as you go further down in the water column. And this also applies to the soil layers. Magnetospirillum likes this because its whole shtick is to use the Earth's magnetic field to orient itself in the water column or the soil column and to move along oxygen gradients until it finds that optimal oxygen concentration. So it's a microaerophile, which means it likes some oxygen, but not a lot of oxygen. This is one of the things that makes it the most challenging to grow. We need to recreate those oxygen concentrations it enjoys while also preserving its magnetogenicity. Magnetospirillum is really well known for losing its ability to form magnets by removing this magnetogenicity island that's present in its genome. And one of the reasons this happens is because of too much oxygen in its environment. For this reason, it's not surprising that it's been found growing on rice roots, which grow in a flooded environment with oxygen gradients galore. It's also been identified in screens of genera of bacteria that have been found in soybeans and maize. Those screens were looking at rhizosphere colonization. So what does this mean for culturing them? Well, there are 2 things to consider right now. The first is the growth medium. So what do you feed them? And the second is the actual oxygen concentration that they need. So I'm going to start with the medium because it's actually probably the simpler of the 2 considerations. I found 2 recipes, one published by the National Institutes of Health. That was really helpful. They went from everything from the setup for the growth medium to the composition of the medium itself and detailed instructions for how to do everything. There's a slightly less detailed recipe which I found which is very similar from Arash Kamali's lab. Honestly, there are a lot of considerations when growing any sort of bacteria, so this media seems to be a mix of as many micronutrients as possible. It's a mix of a wide variety of vitamins known as Wolff's vitamin solution and a wide variety of minerals known as Wolff's mineral solution. So it's basically just a method for growing very picky bacteria. To be honest, I don't know that much more about it. I just know that it works for culturing these bacteria, and so that's what I went with, and I'm going to try it. The second consideration is the concentration of oxygen. So because they're microaerophilic, we need to reduce the concentration of oxygen in the media, and the way that we do this is by introducing a bunch of nitrogen. Air is already 72% nitrogen, or something in that ballpark. I didn't look it up. You're just going to have to trust me. We went with food-grade nitrogen because it has to be sterile for one thing. Food-grade nitrogen is relatively high, somewhere in the ballpark of 98 to 99% if I remember correctly. This is good because it's relatively high, but it's not too high. For one thing, we don't need that purity because we want to introduce a certain amount of oxygen anyway, and it's also a lot more expensive to get very high purity nitrogen. We bubble the media with nitrogen, then we bubble the headspace or the gas layer in the bottle. After that, we're going to introduce a certain amount of air back into the bottle as a way of introducing that little bit of oxygen. So using this method, we can carefully regulate the amount of dissolved oxygen in the media. So now that we've got both the medium and the dissolved oxygen problem solved, we can move on to the next step, which is actually culturing the bacteria. So our lab is reaching out to Arash Kamali's lab, and I really hope I'm pronouncing that right. If I'm wrong, I will correct myself in a future video. So that's the first thing that I've been working on in the past couple of months. The second thing I've been looking at is reproducing the results of that 1996 paper which I found, which looked at the influence of high gradient magnetic fields on root tip growth trajectory. Using ferromagnetic wedges, this is exactly what they said in the paper and provided no further detail. So what I decided to do was make my own ferromagnetic wedges by taking a paper clip and unrolling it and clipping it with wire cutters. Since wire cutters basically have two wedges which meet at a single point, the way that they cut tends to create a wedge shape. With this method, I was able to create a bunch of wedges, but in the 1996 paper, they actually looked at the math that was needed to determine the strength of the magnetic field at a certain distance from the tip of the wedge, and that math involved knowing the exact dihedral angle of the wedges used. I can't guarantee the dihedral angle, and just visually it appeared that I was getting a different angle on one side and the other. But I just wanted to see if I could demonstrate the effect, and so I clipped them and I placed them on agar plates. I think I found some results, but it was largely inconclusive. Paramagnetism is this phenomenon where molecules with unpaired electrons will create an opposing magnetic field in response to any applied external magnetic field. So, regardless of whether the magnet is pointing north or south relative to the material, it will always be pushed away. It'll always be repelled by the magnet. This paramagnetic effect is affecting the amyloplasts more than it's affecting the surrounding tissue. So instead of moving the surrounding tissue, it's just moving the amyloplasts, which is why it's a good method for looking at gravitropism. The consequence of this is that the amyloplasts will deflect away from the area with the strongest magnetic fields. What we would expect as the root tips grow down is that as they approach the wedge tip, they deflect away from the tip. And in many cases, this is what I saw. In many other cases, they seemed totally unaffected and just burrowed their way directly past the magnets without being affected at all. The other thing which happened in a large number of cases was instead of deflecting around the wedge, they deflected underneath and burrowed through the agar to the bottom of the plate and then continued growing from there. Now, the reason I think this is because the cross-section of those wedges was actually circular. So the thickest part of the metal would be the one directly in line with the agar, at least according to the setup that I used, which was to push the wire halfway down into the agar. So if you think of it, a cross-sectional area would have a half circle above the agar and a half circle below the agar. The thickest point is at the actual boundary layer between the agar and the air. So the strongest magnetic field would also be where the thickest part of the metal is. And what this means is that the amyloplasts will be deflected towards the agar. Roots prefer to grow over the surface of the agar because there's much less resistance. Burrowing through the agar requires some energy to push the agar itself out of the way. But because their sense of gravity is altered, they start growing into the agar. I have some other ways I might want to test this, but I started finding that there was metal toxicity as a result of the staples. I think this is from the nickel in the staples, but that's really just nothing more than a hunch. So instead, I decided to use another method which they described in that 1996 paper, which was the establishment of high gradient magnetic fields which can happen between two magnets that are very close to each other. So if you have two magnets and you keep them a certain distance apart, the edge of the field at the gap layer will sort of bubble out, as you can see in this diagram from that paper. Everywhere that is the edge of the gap, the magnetic field is a little bit weaker than it is right directly between the two magnets, because the magnetic field lines end up making a detour. So I tried another arrangement, which was to simply put two very strong neodymium magnets on either side of a square plate and plant the seeds in a line along the top. In this arrangement, there's a gradient of magnetic field strength that occurs as you get further away from the magnet. So you would expect the plants on the outside to be very minimally affected, and the plants towards the middle, towards the center of the plate, to be very heavily affected. From the preliminary results, I think I'm starting to see a phenotype when I add two neodymium magnets. It doesn't really work with one. I'm still going to post it here anyway, but I think this is going to be the setup that I like the most, because it rules out the effect of the solid material itself on the root growth and limits it just to the effect of the magnetic fields. The arrangement I settled on was to make a kind of sandwich with three different square plates, separated by two magnets each. The center plate represents the arrangement that was described in that 1996 paper. The outer plates are there for two reasons. One, it just allows the three plates to stand up straight and maintain a consistent orientation relative to each other. The other purpose of this is because when I was originally testing this, I had a single square plate that had a magnet on either side of the square plate. And I put it in between other seedlings, which I was growing for another experiment I was helping with in the lab. What I found is that the plate immediately in front of the plate that I was testing showed this root tip deflection phenotype right on top of where I would have expected it based on where the magnet was. So this was pretty promising, although the field shape generated by that magnet on the front, I'm less sure of how it looks. I would imagine it's sort of basket-shaped. Two circular magnets will overlap with each other when put together. They won't just sit halfway across from each other. This implies that there's some kind of gradient. But is that gradient a really high gradient? Apparently it's strong enough to cause that effect to manifest. I'm hoping to do some computer modeling, but to be honest, that's a little bit out of my depth, and I need to either learn how to do that or find someone who knows how to do that already and ask for their help. As I get more data, I'll continue to post it on the Patreon, and as I find things that are more interesting, I'll post them in further videos. On to the third part. The third part is actually a genetic engineering approach, which is the original research proposal which I wrote and posted on the Patreon. You can find it there. The initial idea was to take the entire magnetogenicity island, which is made up of 5 different bacterial operons and a total of 31 genes, and move that into chloroplasts, because my thinking at the time was that bacterial translation and transcription was similar enough to chloroplast transcription and translation due to their common ancestry. Chloroplasts, as you might know, are originally most likely descended from cyanobacteria, and they have their own circular genomes, and they're able to transcribe operons. As I thought about this a little bit more and consulted with my PI, I came across a couple of major hurdles. The very biggest hurdle, I think, is that amyloplasts are generally pretty transcriptionally inactive, and so any construct that you put into an amyloplast's genome will probably have a hard time being expressed. The other problem is the transfer of bacterial operons into amyloplasts and plastids in general is not well studied. I tried to find some examples of people doing it, and it looks like, for the most part, it's not very commonly done. In some cases, people have done it with soluble proteins, but very few people have done it with transmembrane proteins. When I got to the step of looking at the individual genes in that magnetogenicity island, I found that roughly most of them were actually transmembrane proteins. And so it becomes pretty challenging if you don't have an established protocol for doing that. The way that I did find that's well known for expressing transmembrane proteins in amyloplasts and other plastids was nuclear expression. So most chloroplast genes are actually expressed in the nucleus and then targeted to plastids based on a polypeptide localization signal at the N-terminus of the protein. So basically, at the end of every protein that's directed to the plastid is a little signal that says, take me to the plastid, or take me to the amyloplast. You can think of it as holding up a sign at the airport with your name on it and where you want to go before you get picked up by a taxi. At the end, you no longer need that sign, so you just chuck it away. And the same thing applies in plastids. That N-terminal signal is chopped off. Me and my PI thought about it, and she came up with the brilliant idea of inserting that localization signal in between multiple genes. Since it's cut off, you would expect that if it's in between 2 genes that have already been transcribed into full proteins, that would cause them to be cut into multiple pieces. So the project I've proposed, and the fellowship application, which I'll post also on the Patreon, is describing a very simple construct involving 1 soluble protein and 1 transmembrane protein for a total of 2 proteins. At the end of the construct is the N-terminal localization signal. In between the soluble protein and the transmembrane protein will be another localization signal, and appended to both the transmembrane protein and the soluble protein will be some kind of fluorescent marker representing 2 different colors. So the idea is to express this construct in the nucleus using agrobacterium-mediated transformation. It's a very common method, tends to be very effective, and is also a lot easier than biolistic bombardment and tissue culture, which is what would be needed for plastid transformation. That's the other issue. Cost, time, and effort. The final consequence of this expression is hypothesized to be that the whole construct will be transcribed on the nuclear ribosomes and then transported by chaperone proteins to amyloplasts specifically based on that sorting signal, enter through this complex called the tick-tock complex, very memeable. After it's internalized, the first N-terminal sequence will be cleaved off. It will just go into the milieu. We don't have any way of visualizing what happens to it. The second thing that will happen is the transmembrane protein will start being translocated across the inner envelope membrane. I'll make a diagram to show you exactly which membrane I mean. Once it's translated across the membrane, we would expect that this internal sorting signal would get cleaved, causing the tagged soluble protein to localize to the stroma of the chloroplast. Again, refer to diagram. We can then use fluorescence microscopy to look at the localization of the two fluorescent labels to see if that's worked. If that does work the way that we hope it does, then you could potentially use the same method to separate 17 different genes with the same localization signal. Then we can use this to move bacterial operons into the nucleus of plants, have them expressed, and then have all of those genes created as a single proprotein, which is then cleaved into 17 different parts after localizing to the amyloplast. So this allows us to use one open reading frame, one transcriptional promoter, and one transcriptional terminator. That also means that we can just use one selectable marker. The advantage of that is that we don't have to introduce multiple antibiotic resistance as part of introducing this construct. Hopefully, we could actually combine all 5 operons that make up the magnetogenicity island. 17 genes make up the minimal system for producing these magnetic vesicles, but the 31 genes are really necessary for full efficacy. We've started the very preliminary steps of working on this project, getting bacteria to grow so that we can inoculate plant roots, and having an assay for root tip deflection in response to magnetic fields so that we can evaluate different methods for modifying that. In 3 months, I've actually made a significant amount of progress. I've also been helping out with other projects as a research assistant, and helping out with those projects has given me an opportunity to make some media and learn some of the basic techniques that are used in this lab specifically, and that has been tremendously helpful. We're going to do some preliminary validation of the method before designing that final construct. I want to create something that is approved for general release so that this is something that people could grow in their backyard or in their garden and take cuttings and share cuttings. And I will continue to update you all on the progress of this preliminary stage of the research. It's been a long journey, and I appreciate everyone who has come along with me for that journey. Special thanks goes to Connor and Sasha for supporting this work through Patreon. I will be back with another sculpture video in the next 2 weeks. This one's going to be a fun one, not plant technology related at all, but very entertaining and some new tricks up my sleeve. So look forward to that. Thank you all so much for watching, and don't forget, feed ducks peas, don't feed them bread. Bread is really bad for them and will give them digestive problems. Peace.